Hi, my name is Arabella, and I will be discussing potential adaptive evolution and pathogenicity in Ralstonia piquetii bacteria aboard the International Space Station. So on the space station, astronauts get their drinking water by recycling the water that they have up there already. These are things like urine, sweat, humidity condensate from the air, and all of this is taken in and compiled into what they call the water recovery system. This uses many different filtration techniques and different chemicals in order to turn this wastewater into potable drinking water for the astronauts to consume. Despite all of those different methods used to purify the water, though, there's been a consistent problem with microbial growth happening within the water recovery system. Over the past 20 years or so, there have been consistent issues with biofilm growth and many different microorganisms found on the ISS. And one of the most common of these bacteria is Ralstonia piquetii. This is a bacteria that is able to adapt very well to harsh environments, such as those with low environments within the drinking water system of the ISS. Um, and this is a particularly interesting bacteria because previous studies have shown that this bacteria is able to adapt to the environment of space. These studies looked at the differences between bacteria identified on Earth and in space, and they found that horizontal gene transfer and adaptation to the disinfectants from the space station resulted in genomic changes and environmental adaptation. Additionally, uh, how However, these studies did not look at the specific locations on the ISS and instead focused on the classifications of space and Earth bacteria. Um, there are different locations on the ISS that routine microbial monitoring is taken from, and these are the condensing heat exchanger, the wastewater tank, and the potable water dispenser, which are just three different parts of the water system that recycles the water. Ralstonia piquetii in particular is an interesting bacteria because it has the ability to cause respiratory illnesses in immunocompromised patients. And this is especially concerning because studies have shown that astronaut immune systems are actually downregulated in space, which means that ingesting these pathogens that are in their drinking water can be potentially harmful to their health and may pose a threat to their immune systems. So as a result, this research investigated two different questions. The first of which was whether the specific location of the water recovery system from which these bacteria were isolated would have an effect on their genetic characteristics. And the second was to take a closer look at Ralstonia piquetii specific genomes in order to determine whether they have potential for pathogenicity and potential to pose a threat to astronauts. To do this, we first collected samples. I worked with NASA's Johnson Space Center with Dr. Wallace and Dr. Nguyen in order to receive bacterial DNA that was collected from the water recovery system directly from the ISS. I also downloaded additional genomes from the GenBank database in order to expand my data set. After that, I performed whole genome sequencing using the Oxford Nanopore MinION, and I also performed de novo assembly using Fly software in order to obtain the entire genomes of these bacteria. And finally, we performed annotation of these genomes using Proca in order to find the specific genes present in each of these genomes. So first, I then performed further analysis in order to investigate these specific questions. The first of which was whether the location of the ISS would affect the genetic characteristics of these Ralstonia piquetii samples. In order to do this, we looked at the pangenome of these bacteria. A pangenome is a sample set's entire collection of gene families, and pangenomes are divided into three classifications. One is the core genome, which includes the gene families that are present in almost every single species and every single sample and are essential to the bacterial growth. On the other end of the spectrum is the cloud genes, which are present in very few samples and are often prone to adaptation and transfer. And in between is the shell genes, which are sort of just at an intermediate frequency. So results of this pangenome analysis gave us this this data. And looking at it right away, we can see that there is this clear distinction between these two groups of the samples. Looking at the blue colored samples here, we can see that they are almost all, or all of them are isolated from the International Space Station. However, looking on the far side at that green, those green relationships there, we can see that the genetic relationships between all of these samples are actually very, very similar. This suggests that the location from which the ISS on the ISS from which these samples were taken from did not actually affect their genomes, as all of these genomes are relatively similar and are very, very close. However, one interesting thing that we noted was that this top section here, these samples colored in red, had hundreds of genes present that were not present in the other samples. And this suggests that there may have been two separate strains or two separate contaminants present on the ISS, as opposed to continued environmental adaptation to the different structural differences in the water system. Looking at this phylogenetic tree, that looked at the core genomes of these samples, we can see the results are about the same. These blue samples here are very, very similar and very closely related, which shows that they are not affected by the different locations. However, there is this group up here that is separated from the rest, that suggests that there was a different contaminant. And what's interesting is that these results also correlate and are confirmed by studies done in the past. Looking at that graph on the right, we can see that the groups that we identified are very similar to the groups that, or almost identical to the groups that previous studies have identified, which means that these strains are basically the same. And this allows us to determine that there may have been adaptation and between these Earth and space environments as found with previous studies, as well as this conclusion of having two initial contaminants, which was also found in previous studies. So moving on to our second question as to whether the genome of Ralstonia pickettii had characteristics of pathogenicity. To investigate this question, we looked at the Phi-based database, which was used to identify genes with increased virulence and pathogenicity. We also performed Proctor annotation in order to determine specific genes that indicated pathogenicity. And we are also in the process of looking at the CARD database in order to identify other genes that show increased antibiotic resistance. So results of these analyses show some interesting things. First of all, we can see that there is increased presence of metal resistance and drug resistance in these bacteria from the International Space Station. And this suggests that this further confirms Ralstonia pickettii's ability to adapt to harsh environments and survive in these environments with disinfectants and with low nutrients. One particularly interesting thing that we found was this presence of a type 3 secretion system. This is a type of protein found on bacteria that allows them to actually inject their pathogenic genes into other cells. And it's an indicator of high virulence, which is a little bit concerning for bacteria that are being actively ingested by astronauts on the space station. Additionally, previous classifications have identified Ralstonia pickettii as being of, quote, minor clinical significance. However, identifying this gene component that codes for this highly virulent secretion system is a concerning sign, and this warrants further research. So overall, this study concluded that from our first question, that the location and the specific structural components of the water recovery system did not have an effect on Ralstonia pickettii genomes. However, we found that there may be two separate strains or two separate contaminants that originated from the International Space Station. Additionally, this study demonstrated that there may be potential pathogenic capabilities of Ralstonia pickettii in the water system of the ISS. Future research may be performed in order to further confirm these results as we are able to look at horizontal gene transfer in order to determine whether there are specific genes that are being transferred from other organisms, other microorganisms in the environment, as well as looking at particular point mutations or SMPs in order to see the specific changes happening in the genomes of Ralstonia pickettii. Finally, we can also look at the specific pathways that are enriched, as previous studies have found enrichment in pathways for different degradation of different chemicals and metabolism of different compounds, which will allow us to further identify and classify the genomes of Ralstonia pickettii. Finally, just some reflections and acknowledgements. Takeaways for future students. Uh, I would suggest being flexible and being open to changes because my project underwent, as Dr. Chan knows, quite a few changes along the way. But being flexible and being open to going wherever the data takes us is a very helpful thing. And I'd also like to thank Dr. Wallace and Dr. Nguyen from the Johnson Space Center for all their support and all of their help in allowing us to do this research in the first place. I'd also like to thank Mr. Hill for his bioinformatics expertise and Dr. Chan for her constant support, as well as everyone else who made this possible. Okay, thank you.